So good afternoon, everyone. Thank you very much for joining us for the second lecture of this mini-series by Dr. Krishna Govender from the CHPC and the University of Johannesburg. Yeah. Last week, Krishna gave us a very nice overview of the whole topic of computer-aided drug design. And today we will enter the nitty-gritties of doing it, yeah, using the Schrödinger software solution that the CHPC kindly makes available to all of us. Yeah. So Krishna, without further ado, you're welcome to share your screen and start your presentation. And maybe a quick reminder to those of you who just joined, we just agreed that we will issue a book prize for the best question of the session, just to motivate you to interact a little bit more with the speaker. Yeah, so please don't be shy with questions, either in verbally or using the chat. Yeah, so this is a seminar, it's not a webinar, so you can ask questions at any time. Yeah, Krishna, please. Thank you very much. I see the screen that you're sharing, and over to you. Thank you.
Hey, thank you, Prof. So thank you guys for joining me again today. So today we're going to try and walk through some of the stuff that you can do with the software package Schrödinger. And this is basically used in drug design. It's currently being used by major pharmaceutical companies when they do the drug design process. Things like people like Pfizer are using it. And one of their biggest investors is Bill Gates, actually, in Schrödinger. He's got a lot of money that he invested in Schrödinger, so just out of interest. So basically what we're going to do today, I'm just going to give you an overview of what we're going to cover. So we're going to install the software, which some of you may have already installed it because you got it from me, and I give you a PDF, which I would have shared with you on how to install it. So those of you that installed it, you won't need to go through this process. I'm just doing it for those that might not have installed this stuff. Then you'll change your working directory, we'll prepare a protein, generate a grid, we'll try and dock the crystal ligand and see how it compares to that of the crystal structure. Then we prepare a library and we'll dock the library. So those are just things that we want to try and cover today. If we don't get through all of it, it's fine. We can finish it up next week together with doing some molecular dynamics. So I'm going to stop sharing this and I'm going to start sharing my desktop. Okay, so you should see my desktop. And what you would have gotten from me is the Schrödinger install file, right? So it's a zip file, which you need to first unzip. So you can just extract it and extract it to wherever you want to extract it to. Right? It's quite a big file, so it takes a little bit of a while to do the extraction. So you can see here, it's going to take like 3 minutes. But while it's busy extracting, I'm going to quickly walk you through some of the steps, right? So inside the folder, there's another folder, and then there'll be a setup. So when the extraction is finished, you will run the setup.exe file. So if I run it now, it will probably run, but it's going to complain. Right, so we're going to get a menu that comes up, which you then click next. So I've already got it installed on my machine, but in your case, you will click on the install option here and you'll click on next. Now, when it asks you for the location to install the application, you need to be very careful, right? So I like using the tools that Schrödinger provides on the terminal as well as through the interface. So if you want to use the tools on the terminal, you should not install Schrödinger in a folder that has a space in it. So on your C drive, typically the first place that you want to install stuff is program files, but program has a space, right? And then it's got program space files. That's a problem because when you try to set a variable to actually point to that directory, it's going to complain to you when you actually run it on the terminal. So what I usually suggest to people is create a new folder, whether it be on your C drive or wherever, and just call it programs, right? Like I've done here. I've got a programs directory in my D drive, and in there is where I install Schrödinger. And I'm going to show you guys how to set the variable and stuff a little later. But I'll install the application in here, and everything I need then goes in there. Now that's just the first step, right? So that's just to get the software installed. It will take a bit of a while to install for those of you that haven't installed it yet. So it's still busy extracting, as you can see. And as I've said, I've already installed it just to kind of speed up our process here. But when you guys are doing it, you're going to obviously go through the video again if you have to, and then you can see how to install it on the machine. So what we need to do next is we need to actually be able to communicate with the CHPC's license. So to do that, you have to go to your C drive, Windows, System 32. So we look for the System 32 folder, then drivers, right, etc. And then there's a file called host. And you'll notice there's no extension on this file. And that's exactly right. The host file should not have an extension. So when you save it, you just need to make sure it doesn't end up with a .txt, because when that happens, it doesn't work. So what you have to do with this file is you need to copy it somewhere where you can actually edit it. You cannot edit it in this Windows folder, so we need to put it somewhere else. So I'm going to take it back to just my D drive in my case. Oh, wait, where did I have it? I actually copied it and pasted it somewhere earlier today. I can't remember where I put it. Okay, wait, let me find it. Oh, it's on my desktop. Yes. Okay, so okay. So I've already copied it and I pasted it in here. Then what you need to do is you need to open this file, right? And we need to point to the CHPC cluster. So because of security reasons, I cannot show you all the IP address on Zoom, and I can't show you the server name either. These are all things that are contained in my PDF document. So when I share the PDF with you along with the installation, you'll find it in the PDF. And those of you using Linux or Mac OS, the step-by-step process on how to install it there is also provided in the PDF. So here you'll basically at the bottom of this file, you'll put in the IP address of the machine that holds the license on the CHPC. Then you'll say the machine's name, .chpc.ac.za, and then the machine's name again, right? When that's done, you just go to File, Save the file, and make sure you save it without any extensions. You then take this file from here, copy it again, and then put it back in the folder where it came from, which is Windows, System 32, drivers, and then etc. And you paste it here. And when you paste it, it will ask you if you want to replace the file, and you say yes. And once it's replaced, then it knows where to go look. So now let's test to see. So I'm going to quickly just test the server. Let me share which screen now. Okay, I will share the main screen. Right, so you can see this. I'm going to minimize that and that and that. Right, you should be able to see just my desktop right now. So what we're going to do is we're going to go to the Windows directory. This is obviously now after you've installed the Schrödinger package, and you type configure. Now I've got a configure Material Studio, which you won't have, but you should see a configure software option. So if you click on configure software, that's the Schrödinger configuration component of the software package. So it's going to take a little bit of a while. It's now going to go and look at that hosts file. From the hosts file, it's then going to go and look on CHPC and see if CHPC has a license. And if it does, then it will pop up with the window. So now it says it's contacting the license server. It takes a little bit of a while. So we'll just give it some time. At this point, if you do not see the license or you don't pick it up after changing the host file and everything is correct, then there's a port that is being blocked, most likely by your university. And it's happened many a time. So I know many students complained about the same thing. They'll follow my steps, but it won't access the license. And the universities usually block that certain port. The port is mentioned in my documentation as well. It's already here. It's 27008, right? That's the port. So that port needs to be unblocked on your university's firewall. And if that happens, you'll enter the server here at this point. You'll just say, okay, I can identify my license server. You'll put the host name and the port, and then you will click view details. And if you see numbers next to each of these things, it means it can see the license. Because as you can see, there's numbers next to each. So there's 200 tokens for ADME, the Bioluminate has 10, and so on and so forth, all the way up till site memory, right? Desmond doesn't show up. I have to still fix that. But anyway, most of the important things that we need to use are all here. So as long as you see numbers, you know it's seeing the license. You can close this, and then you can go ahead and open Maestro. So, Maestro. Maestro 13.1 is what I'm using. This is the latest version of Schrödinger at the moment. So this is the 2022-1 version of Schrödinger. And they've made some improvements to the software and the way certain things are prepared. So it's actually a bit easier to do some of the processes. Right, so let's Maestro open, and I'm going to click check. Oh, okay, so I'm going to change the working directory. So Maestro will do its thing. Eventually it will open. So I trust you guys can all see my Maestro screen now. I see something in the chat. Can different versions provide different results? Okay, so the different versions have different force fields, right? So the thing is, with some of the older versions of Maestro, they were using something like OPLS 2005 or OPLS 2003. And yes, if you go take the experiment that you did in an older version of Maestro and you redo the same thing and redock using a different force field, you will probably get slightly different results. And maybe even in some cases, a slightly different trend. So you have to make sure that you keep the version the same throughout the experiment that you're doing. Try not to chop and change between versions, especially not chop and change between force fields. Because remember, force fields are developed for specific set of molecules, right? If you're gonna go outside that scope, it's gonna give you bad results. And in some cases, it's gonna give you a bunch of notes. So you need to make sure you stay within what you're actually looking for. So in this case, I'm just using the latest version because Schrödinger has made mention to me recently that they've got a new tool which they introduced, which will allow you guys eventually, when I get it up and running, to submit jobs from Maestro straight to CHPC. It will then run on the cluster, and when it's finished, it will come back with your results. I'm very busy trying to figure out the whole process. It's not as easy to figure out, but I will try and get it. In the meantime, I am going to show you all how you can actually get the jobs onto the cluster and run them, because I do have things on the cluster written for you guys, just to make life a bit simpler. Right, so let's quickly go and have a look at what we can do. So this is Maestro, right? So this section here is essentially your window where you will display your molecules. This part here is your workspace. Typically, when you load things, it goes into that workspace. Now, the first thing I always like to do is, by default, your working directory is usually your C drive. And I know most people, your C drive is usually not that big. And Maestro can generate quite large files sometimes. So it's best to change your working directory. That's the first step, right? Just change it to something else. So in my case, I'm going to move it to my D drive. I'm going to put it in a folder called Schrödinger. I'm going to make a new folder. I'm going to call it Mini-School. You will notice I don't use spaces. I'm just a Linux person. Using spaces is usually detrimental. So we try to avoid spaces. Right, so I'm going to select that as my folder. I'm now going to go to save. Okay, we'll save this project later. Now, before we do anything else, let's quickly set up our variable as well. So if you go to your Windows icon and you type variable, you should see the edit the system environment variables, right? If you click on that link, it'll pop up with a menu, which I'm waiting for. Okay. So this is the menu you should see. At the bottom here is your environment variables, right? So you need to set an environment variable for Schrödinger. So we're going to go to environment variables. I've got it set already, but I'm going to quickly show you guys. So you will click on new. Right, let me delete this one. I'll make a new one. Right, you click on new. You then type in Schrödinger in capital letters. And the variable value, just browse for the directory. So go and look for that directory where you install Schrödinger. So in my case, it was D drive program and Schrödinger 2022-1, right? Okay, there we go. Now the variable is set. So once it's set, you can say okay. And okay, right? So now your variable is sorted. I'm going to quickly close this because we're going to use it later. Right, so let me quickly go back to my slides. right? So you've now set your working directory. We've set our environment variable. So what's next? The next thing we want to do is prepare a protein, right? So you want a protein structure, which you can then get from the protein structures, as I've showed you all in last week's talk. The PDB Databank is the best place to find X-ray crystallography structures of proteins. So typically, you're going to use something called the protein preparation wizard to try and set up your protein. Because when you download the protein, it's usually the protein that's crystallized out in a solid state, right? So this is something that a lot of people seem to forget, that the solid state structure and the structure that exists in your body when under the environment of water and everything else is not the same. There'll be changes and there'll be differences in the two structures. So what Schrödinger does is it tries to then make it more in sort of a solvated environment. At the end of the whole process, you want to get it in a solvated environment and see how it performs there, as opposed to just a gas phase thing. So when it does crystallize out, you also get something called an R factor, and you want to try and find crystal structures that have values that are closer to 1. And because the bigger the R factor, then the poorer the crystal structure. So Schrödinger actually has a cool tool which allows you to go onto the PDB Databank and download any PDB structure directly to your machine, provided you connect it to the internet, of course. So we're going to do that now. I'm going to go to File, and I'm going to import a structure. Oh, not import, sorry, my bad. Oh, actually, let's just use the protein preparation wizard from the get-go. Right, so we, okay, so you might not have that shortcut there. I'm going to quickly just close this. I added the shortcut to my window, but usually these shortcuts are not on. They're all under the tasks menu. So if you go to tasks, you should see a protein preparation workflow, or if you want to look for it, just type it out. Protein, and then look for protein preparation wizard, right? You can use the wizard or the workflow. Let's try the wizard first and see what happens. So this is the window that will pop up. Let me try and minimize so it can fit in my screen. I hope you guys can see that. Right. So this is basically the wizard. Now you see here in this block, it's actually looking for a PDB ID. So if you put in a PDB ID, you can then click on import, and what will happen is Schrödinger will go to the PDB databank. It will pull the structure from the databank and bring it back to your machine. So what we're going to use today is, let me just make sure of my crystal structure. 3ERT, right? So it's an estrogen receptor. So I'm going to quickly go back to my PEP wizard, and I'm going to say 3ERT. And the nice thing is it's not case sensitive, so you can do it in capital or small letters. It's up to you. So you just click on import. So you'll see at the bottom of the screen, it says retrieving the PDB. It's reading it, import it. And in the background, there's our protein structure, right? So great. Now you'll see there's also different colors and things here, right? Now, the protein preparation was the old way of doing things. And so we're just using that to import the stuff. What we want to make use of is the protein preparation workflow, because this is the new thing which Schrödinger introduced in the last 2 versions of their software. So this also does the same thing. It can pull the PDB for you. In this case, we already have the PDB, which is fine. You can review the structure. If you want to see for certain defects in the structure, you can then check. In this case, we can see, okay, there's this OHT. What is that? That's just the ligand. So it's telling you that there's a ligand sitting in your binding pocket here, which is good because at least we know where's our active site. But what it also has is waters, lots and lots of waters all around, right? So let's first deal with this before we deal with the waters. So if we use this workflow, you can change things up. If you want the global settings to keep a pH of 7.4, or if you want to use the PDB value, which is pH 7, then you just click this box and it'll use a pH of 7. It doesn't really matter. 7.4 or 7 is more or less okay. You can then also detect the ligands, metals and ions and others, right? So we want all of that to happen. So I'm going to keep those checked. Then you'll notice, as I said, there were red things everywhere, or red blocks along the edges. So what those red blocks say is that are basically side chains that are not complete. So you have certain amino acids which crystallized out, but they don't have their full structure. So maybe there's like an asparagine or something that's sitting there, but it's not complete. It's maybe only got the nitrogen and one carbon. So the chain needs to be completed. So you have to fill in the missing loops. Where on the interface can we see the R? Oh, yes, Darren, good question. I'm quickly going to show you the R factor. Let me go to the PDB databank. Is this the only databank you can use? No, Franklin, there are others. I think Schrödinger has subscribed to other databanks also. And when you say get PDB, if the PDB is not in the PDB databank, then the software automatically goes to any one of the other resources and tries to pull it from there. So it's not just the PDB databank. But let me quickly take you to the PDB databank, so I can show you guys the R factor. Okay, so it was 3 ERT. Right, we search the structure. Right, so there's 3 ERT. Here's your resolution, right? This is the R factor, 1.9 angstrom. So that's not bad. It's a relatively good crystal structure. When you get into higher, like 6s and so, then the crystal is not that well resolved. So usually then there's like a lot of missing side chains and a bunch of other things. And here they also show you there's the chain A, which is then the protein structure. You can also then look at the ligand, and you can have a look at the ligand interaction diagram. So if you click on ligand interaction, you should see a workflow of the different chains, how they sequence together. And then here's your ligand in the middle, and then the amino acids that are sitting around. So I don't really use this interactive tool that much. I prefer using the one that's in Maestro. It gives you the same thing, but it gives you a much easier to read sort of interface. So we're going to look at the same thing just now here in Maestro instead. Okay, so let us have a look, right? So now we have the structure. So we want to fill in the missing side chains. We can cap the termini, but I don't want to cap the terminal atoms because some cases, some of these terminal atoms might be close to your active site. And if you cap them, then it can cause problems later on. And then your hydrogen bonding assignments. Okay, we want to sample the water orientations. We use car to do some stuff for us. The force field that I want to make use of is OPLS4, right? This is the latest force field available in Schrödinger. So that's why I would like to make use of it. And then the distance from the ligand to the ligand to the waters that are closest to it are 5 angstrom. So what usually people do is they remove all the waters from the crystal structure. I don't always like doing that, especially if the waters are sitting close to the active site or in and around the active site. So if I go to my ligand here, I'm quickly going to just display it differently. So those of you that don't know, here at the bottom of my screen, there's the different things, the ligand, the protein, and the solvents. So if I click on ligand and the plus sign, I can change the way the ligand structure looks. So if I do that, I can see the ligand looking a bit different. What I also want to do is I want to, I don't want to kill everything. Okay, what did I do now? Oh, I deselected everything. So let me go back here, and then I show all the hydrogens. Oh, man, come on now. Why is it not working? Anyway, oh, the H's are not put in yet. My bad. We still have to add them in. So hydrogens are typically not on the ligand when you first do it. So they're basically not here right now. We will have to fill in the gaps. So now here's your ligand, right? So you have this whole component, and of course there's things around, there's interacting things around. Now what you can do at this point, I'm not sure whether it works now or it will only work after we prepare the ligand, but let's just try it. So we go to tasks and we're going to look for ligand interaction diagram. Right? You'll see there'll be a pop-up menu that comes up. You just click OK there. Now this ligand interaction diagram shows you these are the amino acids that are sitting around this ligand, which is here in the middle. And at the moment, there doesn't seem to be any interactions taking place. And that's because there's no hydrogens on this ligand, and there's no hydrogens on any of the other things either. So this is why it's important to prepare your structure properly. And then what Schrödinger will do is then add on things like H's for you in the right places. And then we'll look at this interaction diagram again. And a little bit of chemistry now for you guys. Of course, we know there's a double bond if both the high occupancy things are on the same side. It's Z for Zusammen. And if they were on opposite ends, it would have been E. So in this case, Schrödinger is nice enough to tell you, look, this is a Z configuration. So at least you know what isomer you're working with. Right, let's close this. And then to zoom out back to the protein, we just click on this foot view. It takes me all the way back. And there, I can see the full protein structure again. Right, so now, let's see what's next. We want to prepare it. So let me just quickly prepare the structure. It doesn't take long for preparation. So what we can do here at the bottom, you'll notice there are options, right? So there's a job settings, there's preferences, there's write, and there's reset. So if you click on job settings, you can set how many cores you want to make use of for your job. So in this case, I'm going to use 8 on this machine. I'm going to say, okay, because this job doesn't run too long, I'm going to run it locally. We will get to a point where we're going to copy files to the cluster and run it there. So don't worry, we're going to get to that at some point. It's my menu with now. At least, okay. So I will just click on run. Right, so you'll see it will say submitted job. And eventually the job should start running. I'm going to close this window. I don't need that window anymore. So when the job starts running, it will appear here. So it does take a bit of time because it has to communicate with the license server. Come on. There. So there. So you see it is launched. So if you click on jobs and you click on the job that's running, if you monitor it, you can then click on the first one and say monitor again. And it will show you once you actually start getting things in your output file. But this type of computing use GPU or CPU power in case you have an i3 with no dedicated graphics. So Franklin, this part is molecular mechanics. So the cores that I'm specifying there is the CPU cores. So it's running only on CPU at the moment. The GPU part is the dynamics. Unfortunately, Schrödinger's license doesn't allow you to run dynamics on the CPUs. You can only run it on the GPUs. So in that case, we would then have to submit to the CHPC cluster. But we'll get to the dynamics part when we doing that, probably next week. For now, everything I do now will run only on CPU. And of course, I'm going to show you all both the local runs and then a better approach, which is then running it on the CHPC cluster itself. So in this case, you can see the job is still currently running. So we monitor it now. It started doing the preparation. It shouldn't take too long. Okay, but we'll let it do its thing. So don't worry, for some of the longer runs, I've already pre-run the stuff. So I'll be able to show you guys some of the results. So let us check. Unfortunately, the computer I'm using now it's a bit burdened with a bunch of other stuff. So that's why it's taking a bit longer. Now, typically for protein preparation, you would want to do this on your local machine. It's not a very intensive process. So you can run it locally and it should take maybe 5 minutes or so to run to completion because all it's doing is filling in the chains, getting rid of unnecessary waters, and then it'll give you a final structure once it's prepared. So let us see how we finish it. No, it's still running. Okay, so preparing the structure. Okay, so I'm going to give that a second. I want to try and see if I've got a structure that I can import. Okay, if there's any questions, now would be the time to ask me while I'm waiting for this thing. Anything at all? You can see my chat box. So.
Hi, Noah. How are you?
Good.
I'm just speaking to Dr. Swani.
Okay.
Swani.
Yes.
Yes, I've received the invitation to attend the graduations. Yeah, I just wanted to get the duration at maybe on Saturday.
Sorry,
Sorry,
Dave. Sorry, David. I think you are in the wrong meeting.
Yeah, I also think so.
Sorry. Sorry, Krishna, for this interruption. I'm not sure what the question was about. Yeah.
Not a problem. Okay, so Darren, I see not to class pi mole in with Schrödinger, but does Schrödinger run on a language like how pi mole is based on pi? Ironically enough, pi mole is actually licensed by Schrödinger. So if you actually want to get pi mole, you have to buy a separate license, and Schrödingers are the guys that actually provide the license. And Schrödinger is based on Python as well. So a lot of the tools that Schrödinger makes use of, especially on the terminal, is Python driven. So you'll see when we actually run some of the tools, it's like a Python process. Okay, so this is taking a bit of time. Wait, let me see. Maybe I have it here already, then I can just stop this. Import. So protein prep. Right, so there, if the ligand is prepared, that's what it will look like. I'm going to stop this job because now it's taking its own sweet time. But you can leave yours running. It should be done at some point. It shouldn't take too long. It's just out of the interest of time here, I'm going to kill this process. So yes, if Schrödinger does this, don't get worried. It just takes some time to respond sometimes. So leave it until it eventually responds. Don't just close it, because that's bad. What are other functions of the 2D workspace? Okay, so I'm going to show you all the 2D workspace in a bit. So obviously we're looking at everything in 3D now on this interface that's here. But the 2D workspace then allows you to look at the interactions with the amino acids, right? Which we've seen just now from the ligand interaction diagram. But I'll show you some other uses of it now. What else other than water should be there to resemble the biological environment? Typically, the main thing, Mohammed, is waters, but remember the waters that are currently in the crystal is still not enough, right? So after we do the docking, we need to solvate the system completely so that it's submerged in water, so that all of the water interactions are taken into account. So that is something we will get to. Is there a minimum spec requirement for a laptop to run this program? Franklin, yes, unfortunately, they would be. You're looking at something like at least 4 gigs of memory. And typically i3, i5 upwards can be sufficient, but you have to have a 64-bit operating system. Unfortunately, Schrödinger stopped making the package for 32-bit machines, so you have to have a 64-bit. I hope that answered your questions. Right, so here's the prepared ligand, right? Now you'll notice it just says it's 3-ERT and it's prepared. Now what's the big difference? I actually have hydrogens now. Can you see? There's H's all over the place. Let's try and display this a bit better. So I'm going to take the ligand and I'm going to display it with this. Right? So there's our new ligand. So now we can see the ligand. Looks nice and cool. Let's zoom out again. I'm going to go to the protein and I'm going to take the protein and display it as a ribbon. Right? When I display the protein as a ribbon, I want to take away the structure. I don't want to see the nitrogens and whatever it may be. So I'm going to just click on that, on the eye. When I click back in the black window, not the eye, the view, put the ligand back, put the solvent back. So by clicking on this, it just takes away all of the actual nitrogens and everything else. So here is your ligand in the middle of our protein, right? And then you've got some waters around it. Let's maybe also display the waters a bit different so we can see them. So you see when I prepared it, all the additional waters that are sitting outside this active site got removed, the ones that were sitting all around here. But the ones that are close to this ligand, it kept. So because there's possible interactions with the ligand, right? So this is why it kept it. So let me check the chat again. Some proteins come with oligosaccharide conjugation. Is it okay to remove them in protein preparation? Yes. So when you have conjugate ligands, you're gonna have to take it out, right? And especially like with the COVID stuff right now, I know some of the COVID structures that are available on the PDB Databank have co-crystallized ligands, but the ligand is actually covalently bound to the active site. So when you get covalent bonding, you have to break that covalent bond in order for Schrödinger to actually prepare the structure properly. And if you don't break it, you will see you're going to end up with an error. It's going to complain during the preparation process. So that's just one of those things you have to keep in mind. And when you break the bond, it usually gets replaced by an atom like a hydrogen or carbon. So you have to just be careful that it doesn't change up your structure too much from what you're looking for. So these are just things you need to be wary of. Right, so okay, I'm displaying the structure now. Great. Now let's see, what do we do next? Okay, so we prepared this thing. Next thing we want to do is generate a grid, right? So we have the advantage that we have an active site. And because we have an active site, we can actually define a grid, right? So we want our other ligands to actually be docked inside here in our active site. But before we do that, let me quickly do something first. Now you'll notice that everything is one file. And something that somebody asked me last week also, what do I do when the ligand is already bound to the protein? It's relatively simple. Schrödinger is quite cool. You can actually split the molecule. So if you right click on the prepared molecule, you click on split, and then you go into ligands, water, other, right? So now it's split it. So if I click on ligand, it's only the ligand, it's only the waters, and it's only the protein. Great. So I want to take this ligand structure and I want to save it on my machine, right? So I'm going to right click on the structure and I'm going to say export structures. I'm going to save this as original, just orig. I'm going to save it as a MAE file. That's just the Schrödinger format file. And I'm going to say export, right? So now that it's exported, let's quickly check again. We're going to import it back to make sure that we exported the right thing. So I go to the school, I go to original. Right, so now it's showing me everything. I don't want everything, I only want this structure. So sometimes what happens is when everything is highlighted in blue, it actually all gets saved. So we don't want that to happen. We want only the ligand. So we're going to try this again and export the structure. Right, so the project table is actually everything in light blue. The workspace is the stuff you see on the screen. So I should have actually changed that, but now it's fine. The project table, there's only one thing in light blue and there's only one thing selected. I'll export it again and override. Then I'm going to try and import it again to check. Very quickly. Import. Aha, so it's only one structure. Great, that's what we want. Because what's going to happen is when we do docking now, we're going to do rigid docking, right? So what that means is essentially we're going to take the ligand and we're going to allow it to be flexible while we make the protein rigid. The protein will stay fixed in its current position. What if you don't know the active site? So if you don't know the active site, then you would go and use something called site map. So I'll just quickly show you the tool. So site map. Right, so site map will actually help you identify potential active sites. And it usually, I think you can tell it how many you want, but default is 5. And then it will then sample the whole protein and try and find the best positions of possible active sites. And the nice thing with that is you can then use those sites to generate a grid and then dock into that active site. So here we now want to actually use the control key to select more than one molecule at a time. So if you use control and you click, then it selects multiple things. So here is where we want to be. And what we're going to do now is we are going to quickly try and generate a grid. Right, so we go to tasks again and we say grid. So it's receptor grid generation. That's what we want to make use of. So I click on that. Now, here's an option that says tick to identify the ligand. And the ligand's already here in my molecule, right? So if I move this window to the side and I left click on the ligand, there's a box that goes around it. Now that's the outer box. There's actually an inner box as well. So if you go to advanced settings, you'll see there's other options that you can choose from where you can read input structures or specify specific atoms, include H donors, We're going to leave everything default for now. The site is also the same thing. We can change certain options here. And if you go to advanced settings here, you'll see there's an inner box. There's like a green box here. Now this inner box is a 10 by 10 by 10 sphere. We're going to just change this slightly so that it fills up the whole ligand space, right? Usually it doesn't hurt to make it a little bit bigger, but you don't want to make it too big because then it's going to take very long for it to dock. So what it does is it's going to dock the ligand within the green area first, and it will go as far as this purple region when it does the docking. But it will change the conformations of these ligands in such a way that it tries to keep it within the green region. So you see, we're giving it quite a lot of leeway with the purple part. Right, so we picked our ligand. We've changed this to 12, 12, 12, and then we can say run. So again, we can go job settings. We can change this if we want, but there's nothing to change. I'm just going to run it. So we say run. Now, generating a grid doesn't usually take very long, but while that's busy, let's quickly move on to something else. Oh, and one more thing. So I'm going to open MobileX terminal. As I say, I like doing things on the terminal as well. So I'm going to show you why. Because unfortunately, with some of the versions of Schrödinger, there's a little trick that people don't know about, and which I picked up and I've asked them to change. So if I go into my Schrödinger folder, right, I've got my grid file, which is that one. So now if I look at this input file, can you see the force field is wrong? It doesn't allow you to change the force field inside the interface. You actually have to change the force field on the input file and then rerun the job. So here it's using OPLS 2005. But if we started with OPLS 4, we want to have OPLS 4 for everything. So we actually need to change this input file. So just to not go too far, some of those of you that don't know, I'm using Vim here to open the file. You can also use something called Nano if you have it installed. Nano is a text editor. I don't have Nano on my machine, so I use Vim instead. But these are also editors that are available on the CHPC cluster. So I'm going to change this force field to a 4, right, instead of 2005. I'm then going to save my file, and I'm going to go back to Maestro quickly. This job, which is running, I'm actually going to kill it because it's going to give me the wrong thing. So, come here. All right, while it's busy trying to kill itself. Finish. We go back. So the job is busy trying to kill itself. But essentially, there's a script that usually gets generated, which is a .sh script. And this script, this is the reason why you needed to set that Schrödinger variable. Because what it does is it looks for Schrödinger, which is the install place of Schrödinger. In Schrödinger, it's going to run glide, and then it's going to use this input file and everything else. So now that we've changed the input, and hopefully this job has died, yes, we can actually run this file again. So I'm going to run it by just saying /glidegrid.sh. Right? So when I run it, eventually it should start up and run. It does take a little bit of time. But once it starts running, then there should be a new log file that starts to write out, and new information will go into that log file. And what it should then have is the correct force field. So I'm going to cat this file just to see what it looks like. Okay, nothing's being written out yet, but it's running. We have to just wait for a little bit. So while it's busy, let's quickly go back to Maestro. So that's our grid, right? We find out the grid. Now, what's next? If we have a grid, we need to dock something into that grid. Right? So we need to have a ligand that we can dock into the grid. So the first thing we always want to do is, if there's a ligand in the active site, you want to try and dock that ligand in and see if it works. Right? So that's the main thing. We have a ligand, so let's quickly go back to just choosing our ligand. Right, and bring him in so we can check what he looks like nicely. Now this ligand's been prepared with with the protein preparation. But we want to rather try and find different conformers and tautomers for this ligand. So the way to do that is we need to actually use something called ligprep, which prepares your ligands for you. So we go to lig prep. There's the tool. So we click on lig prep. And here we can then put in a few options that we want to select. So we can use the structure. We're not using a structure from the file. We're currently using the structure from the workspace because this is what's being displayed. You can have filters for your ligand. If there's certain aspects you want to knock out, you can always just add some filters. The maximum ligand size that you want is 500 atoms. Force field, again, OPLS 4. We want it between a pH of 7 ± 2. You want to use EPIC for doing some of the conformational sampling. Desalt, generate tautomers, retain this. So all of these default settings are typically for most systems, they're adequate. So we can actually then, right, everything here is set up fine. So we can actually just run this. So it's going to prepare this ligand for us, and then it's going to give us some structures. Now this preparation is being done with a molecular mechanics force field, right? So I'll tell you a bit more. Actually, while I'm on this topic, and since I'm waiting for things to finish now, yeah, let me quickly check if my grid is done. Oh, okay, so see, my grid is done. So grid is finished. It shows you here, it exited, so you know that client is done. So the grid that we wanted has been generated for us. Now the only thing we need is the ligands, so that we can actually dock the ligands. So while that's busy, what you can do is, if you have a single ligand, or let's say 2 or 3 ligands that you want to dock into an active site, one approach to do is use quantum mechanics, right? So you optimize the structure with quantum mechanics, and then you take the optimized structure and try to dock it into the active site. That is one approach. The other approach is also something that Schrödinger has in their newer versions, and which they have in this one also. Once my job is incorporated, then I can show you guys. But it's a quantum approach of finding the different conformations, but it uses a combination of like semi-empirical methods and high-level DFT methods to give you this conformance. So this is called the QM. Where is it? No, why was it? No, back to my slide. Apologies. I need to just remember the name. Yeah. Okay. Oh man, I don't know. It's keeping my mind now. Okay, but anyway, I'll come back to that since this thing is already. But anyway, that approach can actually allow you to then do some quantum mechanics on it. It does take a bit of time because obviously it's doing many quantum steps. So you don't want to have a very large data set when you do that. You rather take a large data set, do a molecular mechanics thing on it, and when you get to a smaller data set, then run some quantum, redock, and see if there's significant changes or not. If there isn't, great. But if there is, you'd rather use the quantum structures then as opposed to the molecular mechanics structures. Is it possible to visualize 2 protein structures and their interactions with the same ligand in the same window, or would you have to run each protein ligand interaction separately? You can visualize 2 protein structures and their interactions in the same window. Remember, there's also, if the proteins are already bound together, then fine, you can view them both and you can view the ligand that's lying between them. If they're not already bound, there's also protein-protein docking, which you can do. So we're not covering that here now, but protein-protein docking is like a whole different aspect. And then in that case, you'd use the bioluminate package for that. So that actually allows you to visualize multiple proteins and ligands. So you can just have a look at bioluminate and see if you can get it done there. Okay, so right, so we have this ligand prepared. Now you'll notice here at the bottom, there's 2 structures. There's that one and there's this one. They're not very different. One has like an H on it. So one doesn't have a hydrogen on this nitrogen atom and the other one does. Right? But conformationally, not very big differences. So let's try and dock these ligands. Since we now have them, we prepared them, let's dock them into our active site. So how do we do that? So we're going to go to tasks and you're going to go to docking. And you're going to say ligand docking, right? So we're going to use glide again. That's the main package that drives all of this stuff. This time, the receptor grid is the grid we've already generated. So we know what the grid is. We can get it from the file, right? So we can browse for our grid, which is there. Right, so that's our grid file. If you want to check it, you can actually see the display receptor box here. And then it shows you there, that's the active site, which is correct, because that's what we chose. So I'm going to switch that off. Then we look at the ligands that we want to dock. The ligands we're docking are not in the file, they're actually here at the moment. So they're in the workspace or the project table, whichever one, because that light blue is also project table. So we want these two ligands to be docked onto this active site. I'm going to keep the settings default. We're just going to use single precision. And it seems like with time-wise, today, this will be probably where we will get to. And then I'm going to show you how to calculate RMSD quickly before we end. And then next week, we'll cover docking more ligands and then also how you do molecular dynamics. So I'm quickly going to run this one. Okay, I'm not going to run it now, but you click on run if you want to run the simulation. I have it already finished, so I'm going to quickly just copy it over. So give me a second. So it was that one. Right, so I'm going to go back there and then I'm going to go look at what it looks like. So I'm going to import my structures and I'm going to go to the docking, and what you want to import is the post view file. So this pb.maedz, that's the file you want to import. You say import and there we go, right? So now what it does is if you click on visualize here, or if you click on this menu here and you say view poses, you can then also view your poses. So there we go. That's one ligand that's docked, and that's the second ligand that's docked. Now you can see very small conformational changes between those two. You can actually hold control and display both of them at the same time if you want to. But now, this is not very useful. It just shows us that, okay, it's in the active site. If we click on the table here on the top, we can get a bit more information. So on the table, there's your R value as well, Daryl. So if you want to look, you can also find it in the project table. So if you scroll to the right, you should eventually come to something called the docking score. And here you will see that the first ligand actually docks better than the second one does, because that's -11 and this is -9, right? So let's have a look at this first ligand and let's see how does it compare to our original crystal structure. So I'm going to select just this one, right? Both light blue and the tick, and I'm going to export it to structures, and I'm going to call this doc 1, right? And I'm going to export this. Let's quickly check it by importing it back to make sure we have the right thing. So there's doc 1. Right, great. It is only one structure, no problem. Right, now we're going to go do some magic on the terminal. So this is where we probably have to end today, but we will do some magic here now. We're in this folder, right? I'm in the folder where I have my simulations. As you can see, there's my files. There's the original, which was the original crystal structure ligand, and there's the dock ligand. Now what we want to do is compare the two. How does the crystal ligand compare to the dock ligand? Keeping in mind that we actually use the force field to try and rebuild this ligand and dock it. So how well does it compare? So the way to do this, let me have a look at the chat quickly. What is considered a good docking score? There is no such thing as a good docking score. To be honest with you, the docking is all dependent on, you don't want positive values, obviously, for docking, because then that means there's no enrichment whatsoever. Because Schrödinger just does an enrichment process to actually find the docking scores. It bases it on things like hydrogen bonding, van der Waals interactions, and so on. And you'll see when we do the ligand database that docking scores can vary considerably. You want the docking score to be more negative, but you just need to also look at the interactions that are being considered. And there's certain workflows that Schrödinger give you access to, which would then pre-process some of the stuff for you beforehand, which is advantageous, right? So we'll have a look at one of those workflows also next week. So let's quickly have a look here. If we see I want to compare this structure to that structure. So what I need to do is I need to use that $Schrödinger variable. And okay, wait, let me just quickly check my history. I can't remember the command. Yes, right. So I say $Schrödinger run.exe. Then I want to calculate RMSD.py. So this is why I say there's a lot of Python stuff that's already built into Schrödinger. So to calculate the RMSD, we want to calculate it between the original and between doc1. It's busy running. It shouldn't take too long. Right, so there we go. So there's our RMSD, 0.75. So not too bad, right? I mean, considering that we're now using a force field to read doctor's ligand. And if you visually go and look at it as well, let's pin this. Double clicking on a protein will pin it to the window. So if it's pinned, anything else you click after that will just go inside this protein. So if we look here at this ligand here, which is the 11 point something, and we compare it to our original ligand, which is up here, and we zoom in, see, it's not a massive difference in conformation. Only here is where the biggest change seems to come in. So, but not too bad, not too shabby. Right, so this is basically the whole docking of a single molecule if you already have the molecule in the active site. And of course, the fact that we now have a grid, it's advantageous, right? We can use that same grid to generate, to take other ligands and dock it into the same active site to see what we get. So what I will do is, I'll quickly just show you, because we've got like 3 minutes. Let me quickly go to the PubChem, right? But before we do that, let's go back here. Now the cool thing with this is if you have the ligand, let's go back to our original ligand. Oh yes, another thing is, which I forgot to show you guys, is you can look at the ligand interaction diagram, right? So there's your ligand interaction diagram. You see there's glutamine 353 and arginine 394 that are hydrogen bonding to this OH group here. And surprisingly, those waters that are there are doing nothing at the moment. So that's quite interesting. But they are quite close, but I don't see any hydrogen bonding being formed. So let's quickly have a look at the original ligand on. So that was the docked ligand. Then this is the original one, right? On the same active site. Same thing, glutamine 353, arginine 394. And similar process, there's no waters that are actually hydrogen bonding. The criteria for parameters when you do a study, for example, a set applied of guidelines you must use for a study. No, there's actually not. What's usually helpful is that you go to papers and check what they've done and try and build on what they've done in the paper, and then obviously expand on it later. So, and most of the time, using the default parameters in Schrödinger actually works quite well. I would suggest not to change them. It's only if you see certain funny sort of interactions happening, then maybe consider changing it upwards. As the last thing, as I said, I wanted to quickly take you guys to PubChem, but I need to first select my protein, look at the ligand interaction diagram, right? So you'll see this is just the protein now, nothing else. And the cool thing is we can actually export this to a file. So I can export this. I'm just going to put it here. I'm going to call it structure.stx. Fine, we'll save it. I'm going to close this, go back to the website. I'm then going to say draw structure. And at the bottom here, at the bottom, you will see there's an import option, right? So you can choose the file. Quickly go here to my file. Oh, Schrödinger Mini-School. And then I will choose structure.stx. And then I say import. So it takes a little bit of time. Sometimes you have to click it more than once also. It's doing something. Okay, I think the structure is there. Sometimes this website is funny. So let's search for it. So you see what it does is it does a smile search, right? And there it's got, okay, it didn't do it properly because it didn't bring the structure properly. All right. So, okay. Come on. I won't be much longer, guys. Just want to show you all this one part. There. So you see now when the structure is fully loaded, this is what the structure looks like. And we can search this website for a structure that looks like it or structures that look similar to it. So in this case, it's busy finding stuff. It didn't find the actual structure itself, which is surprising, but it did find 1,000 similar structures, right? So it's found a bunch of different ligands. And if we say search all, it will give you the full 100%, right? So it's now 1,052 ligands. And what you can do is then you can then just download the 2D structures of this. So you download and you just say download 2D SDF. And there you should see the download starts. So I want you to just download this. And what I want you to try is set up these ligands for docking and dock them to this active site. And then see what happens. Right, so how do you rotate around the protein with your mouse? Oh, okay, apologies for that, I forgot to tell you. So rotating the protein, there you need to use a mouse, a proper mouse. You can't use your mouse on your laptop because it doesn't work well. You actually have to then just use the middle mouse. So you see the button that you use to scroll in and out with? If you click it down, it actually clicks. And that allows you to rotate the molecule along the XY axis. So the same one that you use to zoom in and out. Just click it down and then turn. Because if you do that, you can't turn it. And you can't turn using the right click either. You have to use the middle mouse to turn it around. Right, so let me just stop sharing here. And I know we didn't get to the CHPC part, but I think next week we will then do that just to show you guys how to do the CHPC stuff.
Yeah, Krishna, thank you so much. That was again a fantastic lecture and a great introduction to using Schrödinger. It's fascinating, all the things that you can do. Yeah, and I guess we have around 50 participants that will play around with it until next week, and hopefully we'll do the homework that you suggested. Yeah, thank you. Thank you so much.
And I want you all to see how long it usually takes for the 1,052 ligands to dock into the active site. But remember, you must prepare the ligand first before you dock it. So that's one trick there.
Okay, excellent. Krishna, you were really fantastic in answering on the fly all the questions in the chat. I would just like to give the participant one last opportunity for today to ask urgent questions. Please either unmute yourself or use the chat. And what I will do later, I will send Krishna the report of the chat, and he will let me know which question he thinks was the best one so that we can award the book prize for today. Are there any more questions? Or is everybody already busy playing around with Schrödinger?
There was one last one. What is the software to use for running the comparison between the original
I didn't see that. Yeah.
and docked ligand and get the docking score? So Schrödinger does it all for you. You literally just need to go to the project table. So on the top there, you'll see a table. And when you click on it, it then shows you the things like the docking scores and everything else. So you can get it all from there. How about cases of model structure where there are no co-crystallized ligands? In that case, you're going to want to use something called sitemap to actually identify active sites for you. And then you use those sites to dock the ligands and see how they perform.
Excellent. Fantastic. Thank you very much. Sorry, there is one last comment, Krishna, for you.
The RMSD within the terminal, yes. Do you want to expand on that?
It was more a statement than a question. Yeah. Here comes the question.
Best way to self-learn docking and MD. The Schrödinger tutorials, they're great. So if you go to Schrödinger's website, because you'll have access to the software, you'll have access to all of their material. And you'll see some of the stuff I'm doing here are things they cover in their tutorials as well. And they do like 15, 20-minute lectures on different pieces.
Excellent, fantastic. And maybe just as a preview to the participants, Dr. Govender kindly agreed that if there is interest after this introductory course, we can organize a more advanced mini school later in the year. Yeah, so please let us have your feedback. And if we see that there is a critical mass of participants interested on learning more about Schrödinger, we will be able to convince Krishna to offer us another mini school at some later stage. Yeah.
I see there's one thing, sorry. Can you do many other Schrödinger stuff in the terminal? Yes, you can do a whole lot of stuff in the terminal. So the advantage of this is that when you're running it on the cluster, see, like with the RMSD, for example, you don't have to now bring the structure back to your machine, to the RMSD. You can do it on the fly, on the cluster, and then work out your RMSDs and only bring important stuff back to your machine. So there's many things you can do. There's lots of Python scripts in Schrödinger. Would using Schrödinger give better docking results as opposed to using autodock or other? Mullah, no. To be honest with you, each software uses its own algorithm. So they all have their pluses. And autodock is actually quite good. It's open source, but a lot of people make use of it. And it's quite good at doing the docking and docking predictions. And it doesn't come too far from client, which I've seen. Good for chemists will be much appreciated. Oh, okay, that's good for chemists.
Yeah, no, we will do that. We will do that. Okay, now I think the thank yous are starting in the chat, Krishna. So there are no more urgent questions, and next week there will be another opportunity to ask further questions to Krishna. So thank you very much, Krishna. It was again fascinating. Thank you for the many participants that were with us this afternoon. Don't forget to join us next week. And if you are in the Easter Club, all the best for Easter. And we are looking forward to see you again next week. Yeah, thank you very much, Krishna. I will send you the chat so that you can choose the best question of today.
No problem.
Thank you. Thank you very much. All the best, everyone. Cheers.
Thank you, guys.